Kia ora koutou, ko Rebecca Swan Tukuingawa. I'm one of the co-founders of Growing Point, and we are based at Dignam Street Community Garden in Point Chevalier. And today we're going to be running a workshop called Exploring Microbes in the Soil and How They Sequester Carbon. So Growing Point has just recently become a certified soil food web laboratory, and it's very exciting to see all these little critters, and we're going to be showing you that through the microscope, and so you can see the life in the soil and that relationship that they all have with each other. So I hope you enjoy it, and share this with your friends, and here we go. What we're going to do is a very brief intro in terms of the biology and the 4 different groups that we're going to be looking for, and going through identifying each of those 4 groups, and then and we'll do how to collect a compost sample, and then how to make up a drop, and then we'll put it in the microscope, and then we'll do a live view of what we're actually looking at. So that's the plan. And hopefully you'll get as excited about looking at these tiny little things as I do. It's been quite a life-changing learning for me, especially the connection between all life. It's a very beautiful kind of metaphor for how we are all so interconnected, and this is a one thing that we can't see with our eyes, but it's going on all the time, and the scale of it just blows my mind. If you've got healthy soil, there's more microbes in a teaspoon of healthy soil than there are humans on the planet. And we're going to be looking at 1 drop, and it looks like Manhattan from the stars in terms of the amount of life that is going on in there that we can't see, but we feel it. We feel the energy of it, and we eat it through our vegetables. So it's awesome, your questions about wanting to improve the soil, which will then improve the food that we grow in that soil. So I will share my screen. Find my messy desktop. So just a quick overview. Can everyone see that okay? All right, so this is the basis of this relationship. Before we look at the actual microbes themselves, the relationship is all the connections between these different elements of our ecosystem. So the sun creates the energy, obviously, that then the leaves are photosynthesizing. The plants themselves then gift those sugars, those exudates, out through the root system around what's called the rhizosphere, so the area around those roots. And then these sugars are fed on by the primary consumers in the soil, which are the fungi and the bacteria. So those are our first 2 primary decomposers, and they're feeding around the root system. So the plant is the engine to feed this whole system. So you can make beautiful compost and have beautiful soil, but if you leave it without plant roots, the soil biology goes very quickly. It needs the engine of the plants to be feeding this whole system. So that relationship moves into our fungi and our bacteria. And then these are consumed by the next level up, which are our nematodes. We've got 4 different types of nematodes that eat different things. So they've got different mouths because of what they eat. So we've got bacteria feeders. I'll go through that when I show you the nematodes one. And we've got our protozoa, and then we've got the arthropods, macroarthropods and microarthropods, and then we've got our earthworms, and then our larger animals that consume that. So what we're going to be looking at today is the 4 groups and their relationship to each other. Oh, it's not letting me zoom through here. So the bacterial group, there's different types of bacteria, and we're going to, they're the smallest in the whole group. And so we're going to be looking at basically the bacteria en masse, but there are bacteria that are indicators of good soil and compost, and that are indicators of an anaerobic system, which are these bacteria in through here. So predominantly we'll probably be looking at cocci and bacilli, and that will have a combination of those types of bacteria. If we start to see a lot of spirochetes or spirilla, that is, it's actually human pathogens, and we don't want to be dealing with that without washing our hands very carefully. And it's it's an indicator that your soil is in a bad situation. So then we go to our filamentous strands. So the fungi isn't the only filamentous strand. We've also got what's called actinobacteria, which is a bacteria, but it's in a strand form. So we need to differentiate between what we're looking at in the strand form. And we've got a range of great fungi, good fungi, okay fungi, and bad fungi. So we're hoping to see some nice strands of our beneficial fungi, which generally are dark brown. They're wider in diameter, and they've got septa walls partway along that filamentous strand. These are our nematodes. It's tricky to see because they move quite quickly, and so you've got to diagnose generally what their mouth shape is like as to what type of nematode you're looking at. Predominantly, we'll probably see our bacteria feeders, which is in this one here. The fungal feeders have got this little spear, and that's for piercing the fungi and consuming it that way. We've got our root feeders. We don't really want root feeders in a system because they're going to be eating your plants, your plant roots, not in a good way. We've got the predatory nematodes. I've only ever seen one of these. They've got this massive tooth and a big mouth. So they eat the other nematodes. And then we've got the omnivore, which I haven't seen any of these. So predominantly we'll be hopefully looking at some bacterial feeding nematodes. And this is looking at our protozoa. So we've got 3 main groups of our protozoa. We've got flagellates, which have got a little flagella, sometimes at one end and sometimes at 2. And they're good indicators. They eat a lot. Protozoa eat like 10,000 bacteria every day, one protozoa. So they're going to be changing the balance between our bacteria and fungi relationship because they're consuming so much bacteria. Then we've got our ciliates, which they're called the racing cars of the soil. They zoom around. They've got little hairs all the way around them, so they can move in any which way and very fast. And they're bigger than a flagellate. And then we've got our amoeba, and we've got them in different forms. We've got a naked amoeba. We've got our testate amoeba, which we will definitely see some of those. And then we've got amoeba cyst, which is in a dormant form. So we don't count things that are dormant. We're just looking at the elements that are live. So just to give you a brief context of what we're looking at, Tim and Gaia came along. Tim has made this, through the Carbon Cycle Company, has made this structure for us, which is a prototype of their infrastructure of their bins, but using a much more open mesh so that we've got more airflow around it to keep the system really nicely aerobic. And these sides come apart in 3 stages. And this is what we've built, the compost that we're looking at in this infrastructure. This is an earlier one that we built just in a cage. So you can do it in, you know, hardware garden with materials that you could get from a hardware store. The key is the methodology and the recipe. I won't go too much into that. Well, we can do that possibly in another session. But yeah, this is what we're looking at, and it's a very young pile. So I haven't got the exact date, but it's about 5 to 6 weeks since we've built it, and the biology in it will change all the time. From week to week, it will be changing because everything's eating each other. So I'll now stop sharing and show you how to take a compost sample. And this is what I collected yesterday. So this is what the compost is looking like. It was just after that really heavy rain. And so it is at a higher moisture level than it was when we built it. So it's probably around 70% moisture. And it's interesting to see the biology change because of that moisture level. And when I collected this, I got it from 5 different spots in the bin so that you're getting a representative sample. And I just put it in a Ziploc bag, sample it up. And before I take the sample, I just squish it around a bit to break up those aggregates so that it's releasing the biology inside of that without breaking it up too much. So that's the sampling procedure. And then I've tipped that out into my little dish that I again mix through so it's a bit more homogeneous. You can still see it's very recognizable, the inputs. It's still quite twiggy. It hasn't broken down very far, but this is a really good example of what might, to your eye, look like, oh, it's cruddy compost. It just looks really sticky and woody. But actually, the life in it is pretty impressive, I have to say. So once you've collected your sample, you then put 1 milliliter into a test tube, which I've done earlier, and top that up to 5 mils of non-chlorinated water. So I've got a carbon filter on my hose in the garden. If you go to all this effort to create biology and then you use chlorinated water, if you use chlorine, it's just going to kill all your biology. So you don't want to be doing any tests with water that's got chlorine or chloramine in it. So this is a carbon filter water source that I've made this up with. You then do a very consistent shake, which I have done on the sample, but just to demonstrate, I'll move this around. So I'm going to move my hand from, if it's a clock face, from 12 o'clock to 9 o'clock, 30 times like that, and about once every second. So you do that for 30 seconds. And what that does is release the microbes off the substrate, or the organic matter, and into the solution. So you don't, so we can then look at it through the microscope. So this is the mind-blowing bit, right? So that's 1 ml of our compost sample. We're then going to take 1 drop of this 1 ml from our solution. So after you've done your shake, you wait. This is quite a dense liquid. You can see already the color of that is a nice dark chocolate brown, which is an indicator that we've got good presence of humic and fulvic acids. And that's the coloration. So you can do, if you don't have a microscope, you can do that test in a jar with some water, shake it up, let it settle, see what color that your solution is. If it's got, if it's dark brown like this, we know we've got humic acid in here. If we've got humic acid in here, we've got fungi in here. If it's kind of honey colored, we know we've got fulvic acid, which is sort of the precursor to humic acid. So you've still got some good activity. If it's a lighter brown, if it doesn't color up at all, then you've probably got no fungi at all. So that's sort of a test, a non-microscopic visual assessment that you can do. So I'm now going to draw up 1 drop in my pipette. I'm going to sit underneath the level, and then I'm going to release that back into the test tube. This is because microbes stick to surfaces. So I've now coated my pipette with microbes so that it's not going to skew the results. And then I'm going to draw up a 2nd drop. And then it's going to go straight on my slide. So that's the scale we're talking about, like 1 drop of 1 ml. I'm now just going to clean my pipette. You don't want to leave that unclean, because you'll get a biofilm buildup on your pipette. So then I've got my, it's a 22 by 22 cover slip. It's clear, so you probably can't see it, but that's it. Maybe you can see. There you go. And off my drop, I'm going to drag the drop across the slide with my cover slip to coat that whole area that the cover slip's going to drop on with the microbes. And then I put it underneath my microscope. So I'm just going to flick to sharing my screen now so you can see what's going on in on that slide. So in the microscope, I've got 5 different lenses, which are different magnification amounts. So this is my 10 times objective lens. This is my 4 times objective. So generally, to start a sample, you want to have a look at sort of the widest field you can, and then you zoom in to start identifying the different microbes. So this is on the smallest. It's a 40 total magnification. So we can have a bit of a look. It's quite dense. There's a lot in there, and we can move our slide around and just have a general look-see. We can see there's some filamentous strands in here, but until we go closer up, we won't be able to identify them. So we'll just do a general look at our sample, and then we're going to up our magnification to be able to identify the different groups. So the 4 groups we're looking for: bacteria. I've never seen a sample that doesn't have bacteria in it. You don't have to worry about your bacteria. You do though want a diversity of types of bacteria. If it's all one type, it's not a great indicator. You do want to see different varieties of the cocci, the little dots, and the bacillus, which are the rods. And you may find some of your other ones that are not great indicators of what's going on. So I'm going to hone in on that little cluster in here and see what we can find. Oh, boom. Okay, so we've got life, a lot of life here already. This is just on our 100 times magnification. And to do full IDs, unless it's quite a large sample or specimen, you need to go up to the 400 times. So we'll do that. And take a look at what we've got going on here. Okay. So get my focus in. Now, what looked like a filamentous strand that I got a bit excited about is not fungi that we're looking at here. And the reason we know it's not fungi is because the diameter isn't equal all the way along. It also has no septa or walls across the fungi. So that will be just a piece of organic matter. However, in this, it's called a field of view. We can see this is a protozoa in here. And that's a testate amoeba we're looking at there. And they create a little shell around them as protection, and then the squishy bit of the amoeba is on the inside. And you can see there's a double wall there. So that's a testate amoeba. Sometimes they're shells without anything active, so you want to check that there's actually a living thing in there. So that is an alive testate amoeba. So that's in our protozoa category. All the little dots swimming around there are our bacteria. So they're the smallest group, and they move quite quickly. A little sort of shimmer, just sort of like a vibration. And we've got another testate amoeba in here. And these little clumps here, that's organic matter, and that's showing a little aggregate. So when they clump together like that, and you can see this sort of honey color, so that's an indicator that we've got some fulvic acid in here. So I'm going to go back to our 100 times total magnification and have a little scoot around and see if we can find an example of beneficial fungi. I'm just going to do that through the eyepiece, and you get more clarity with the eyepiece than you do with the camera view. So while you're looking at all this, just remember this is 1 drop of 1 ml. It's and there's so much life in here. Okay. All right, so we've found a strand of beneficial fungi. And because of the drop, the depth of the drop, you can, you need to focus all the way along the strand to check the diameter, because what looks like it might be sort of skinnier at that tail end when you focus through on it, is actually the same diameter. We've got a little flagellate just swam past. I don't know if you saw it on the screen. I saw it in the eyepiece. And it's a dark brown colour, which is an indicator that it's some good beneficial fungi. We've got more testate amoeba in here. So we've got one here. We've got one that's dividing in here. So when they divide, they create a new test and then they squidge half of their stuff from one to the other. It's quite exciting to watch. We've got a testate amoeba here. Lots and lots of bacteria through here. So as this pile matures, as I said earlier, one protozoa eats 10,000 bacteria every day. So you can see there's a lot of protozoa, there's a lot of amoeba, testate amoebas in here. They will be consuming the bacteria, which will lower the bacterial population, which is, as the pile matures, you want that to happen. You want ideally, you want more fungi in the pile in relation to the bacteria populations. So I'm just going to try and find us a nematode, because they are the fun. So we found 3 out of our 4 groups. We've had fungi, protozoa, bacteria, and the last group is our nematodes. So I've gone down to 100 times magnification to scoot around and see more of the field. Okay, here's a nematode. Woohoo! I would have felt like I'd overpromised and under-delivered if I didn't find us a nematode. So this is, if you've heard of like nutrient cycling and the poop loop. So this is how the poop loop works. It's through our nematodes that eat a whole lot of bacteria. Can you see it there? That nematode is full of bacteria in its insides. And so, and then it poos out the excess carbon in a form that the plants can then take up. So this is how the poop loop happens, through the predators eating up the bacteria and then pulling that out in a form that the plants can then take up. So if you've got a good biological system with all those 4 groups operating well, you will have enough nutrition from your soil, because it's the biology that's releasing that. I'm going to try and find this. Other interesting things to look at. Okay, here we've got another nematode. You can see it's thrashing around. I can see it more clearly through the eyepiece. There's a little bit of lag on the video, and it's harder to see through the zoom. But it's thrashing around and it's eating up the bacteria that's sitting around this aggregate of organic matter. So when I'm doing a full analysis, I'm going to count all those things. I'm going to count that bacteria, if you can believe it. I do it in a massive dilution. So you do like a 1,000 times dilution, and we've got a 1 to 5 dilution here. And you count the bacteria in a particular area. You count the nematodes and you identify the type of nematode. You won't be able to see it from the screen there, but I'll look at its mouthpiece. And that is a bacteria feeder. So it's chomping through a lot of that bacteria, pooping it out. So then the plants can then utilize those nutrients in the poo to get fed all the nutrition it needs. I'll just do another zoom around and try and find another good example of a fungal strand. You can see it moving a bit more clearly in the 100 times magnification. So I had a look at this last week, the same pile, and I was really impressed with the amount of fungi that has come on and the size of the strands in a really short space of time. And I would put that down to the soaking that we did. So we pre-soaked our carbon content on this pile. It was supposed to be for 2 weeks. That was off an innovation by Paul Stamets, who is if you like mycology, just check him out. So he's done a TED talk, but this one was an example of pre-soaking your carbon to get your fungal activity really kickstart. So this is a beautiful strand, gorgeous bit of fungi here. It's dark brown, so it's an indicator that it's a beneficial fungi. It's parallel sides all the way along. If we focus through that strand, you can see the septa walls really clearly. So generally those are evenly spaced. And a fungal, to differentiate between a fungal strand and a bit of organic matter or plant root, they have very sharp breaks on the ends. So they're never kind of raggedy on the ends. And I would put that diameter at about between 4 and 5 micrometers wide. So the bigger the diameter, the more beneficial that fungi is. So that is a pretty nice example, and you can see it's starting to branch off. Now, this is part of EcoFest. This is carbon sequestration in front of our eyes. This really excites me. So basically, the walls of these fungal strands are storing our carbon from the atmosphere. So the more fungal activity we have in our compost and our soils, the more carbon we're sucking out of the atmosphere and locking up in our soils, sequestering that carbon. And to me, this is my whole kind of drive and passion for this, is that this is the potential, if we can create these healthy soils and soil food web with all this activity and a good fungal proportion in it, we are sucking carbon out of our atmosphere and locking it up in the soil. And on that note, let's have some questions, because I've been talking for far too long, really. So I'll stop sharing. And there's no specific questions in the chat yet, so let's just open it up. Who's got something?
Bex, can I ask what you soaked your carbon material in?
Yeah, it was just rainwater.
Okay.
So you the idea is that it goes anaerobic, which is a little counter to a lot of Elaine's teachings. However, I did ask her, and she says if it's good enough for Paul Stamets, it's good enough for her. So what happens is that you will be breeding your anaerobes, which so the bacteria will go off in that water-soaked environment because there's no oxygen in there. And then at the point that we drain it, so as soon as it gets exposed to oxygen, that bacteria will become dormant, but it's fungal food and the fungi goes off on it. So we've done other piles where we haven't done the soaking and we haven't had the same explosion of fungi in the same way that we have in a pile so young to see that degree of mature fungi. I'm putting down to that pre-soak. Paul says do it for 2 weeks. We ended up doing it for 6 weeks just because it was over the holiday period and we didn't get to build it in time. So we're just accumulating more materials to do another trial of the same recipe, but we'll do a 2-week soak instead of a 6-week soak and see if there's any difference.
And would you do that with all carbon material, like leaves, wood chip, everything, or more dense carbon material?
Good question. We are doing it with all our carbon for this one and seeing. Paul's experiment was just with very woody wood chip. You know, it wasn't fine twigs or anything. Yeah, the theory is that it's the breeding of the anaerobes on that's going to explode your fungi, which is going to. And what's also been interesting to observe is that what started off looking a very woody pile has started to soften, and so those harder woods are being consumed because it's the fungi that will be consuming those harder woods that the bacteria and the protozoa can't consume. Yeah.
Great, thanks Bex, love it.
We just did it in wheelie bins. And actually kept some of the liquid that we tipped off, that was actually Tim and Gaia rolling those through the garden and draining them. But we also kept some of that because it will be full of that anaerobic bacteria if we needed further moisture in the pile to utilize that liquid that's going to be the food for the fungi as well.
Cool. Thanks.
Pippa's put in, Kiss the Ground is a fantastic documentary film about regenerative agriculture and soil's amazing ability to sequester carbon. It is awesome. Anyone else got specific? Would you like me to go through the recipe? We're about to post a video on our Growing Point YouTube channel about the build of this particular recipe, but I can just say it briefly. It's 50% carbon, and we had a mix of wood chips, already made compost, because that becomes an inoculation for the next pile, and some coffee chaff. So we had those 3 materials that made up our carbon, but the total carbon by volume was 50%. Our nitrogen, which was basically weeds from the garden trimmings, 30% nitrogen and 20% high nitrogen, which is considered the party food. So that's what's really going to ramp up your temperature. It's going to feed your bacteria massively. You're going to have a bacterial explosion at the beginning and spike your temperature. And so we did freshly cut grass. So we're really interested in just using materials that are really accessible for most sites. So it was cut grass, weeds and trimmings for your nitrogen, and woodier material, which we had arborist mulch and the compost and the coffee chaff. So 50% carbon, which is heavier in carbon than normal, and that's where you're going to get your fungal activity with that carbon. 30% nitrogen, 20% high nitrogen. You can use cow manures for your high nitrogen as well, but we just wanted something that was more accessible to more sites as a spore. Okay, there's a few questions in the chat. Ali says, you don't use any animal inputs in the compost, do you? They are not needed. No, we haven't on these. Do rust spores live in soil? I have a recurring issues in my garden and worry about what goes through my compost. Is there a way to help prevent it at a soil level? I would say, I don't know exactly. Judy may be able to answer that better. My thinking is that if you've spiked in your temperature, that could kill rust spores. Do you know, Judy?
I would say put it in. The only thing I don't put in is blight infected material. Anything with spores. It does seem, and Daniel was the same on that. His input was saying that the microbes would resolve the issue through composting. Yeah.
So we, the methodology in this particular recipe is that we want it to go over a certain temperature for a certain period of time. So either over 55 for 3 days, over 65 for 2 days, or over 74 for 1 day, 24 hours. So we're monitoring our temperatures very carefully after a build, and making sure that that happens, and we do it, it's a 2-turn system, so that everything gets a turn in the cook zone, which is the middle section. So we do 2 turns, and every turn we've made sure it's gone through that first spike, the thermophilic phase, so that we know all our weed seeds are killed, because it's full of weed seeds. That the material we're using is full of weed seeds, but we know that they've been cooked off and they'll be not viable in our end result. Is there party carbon as well? I like that idea, party carbon. I think the party thing is that it's just that kick-starting, but I guess by soaking it, it becomes more active more quickly. So we could call a pre-soaked carbon party carbon. I don't know. Let's just make it up. But yeah, that would, I guess there's carbon that's more easily digestible. So the softer things that you can kind of crush and crumble with your fingers, twigs that have dried out, will be more quickly consumed than the harder woods. Pippa asked, does using antifungal sprays on plants negatively impact the fungi in the soil? Absolutely. We do not want any fungicides, pesticides, herbicides, anything with sides on the end of it is like death to microbes. So we're using the biology to create the immunity rather than these sprays. So if you have healthy microbial system with all these 4 functioning groups, you are going to have healthy plants that are disease resistant. That's the idea. So anything that's and a fungicide, even the organic ones like neem oil, a lot of people will use, but it's a broadcast. It's going to kill everything, and so you're going to kill all the good guys for the bad guys. It's like antibiotics in people. We want kind of immune responses from the plants themselves rather than something over the top that might be a quick fix, but at the end of the day, you're going to kill that soil food web. You're going to kill that in your soil by using those sprays. How do you measure the temperature? We've got a very lovely long compost thermometer. Tim, you sell them, don't you? So the carbon cycle companies sell the long compost thermometers. I have used a milk thermometer before I got my long one. Clean it well before you then stick it in milk. But yeah, I mean, you'll see it too if you don't have any thermometers. You know, when you're up to 55, you've got, you know, vapor coming off it. It's super hot, but yeah, a thermometer is a good idea. So go to Carbon Cycle Company if you want one of those long ones. Any tips or main signs you look out for about the ecology in the bin without using a microscope? Yeah, good question. Yeah, so you want to keep an eye on, well, the temperatures in the beginning. You want to, the moisture level is really critical. So we did have a pile that we made all, you know, with all the different methodology, and it dried out over summer and the biology just really dipped. So we're going to be much, because we're not using food in our systems yet, the idea is that we will move to that once we've nailed the basic recipe. But we don't have as much moisture content in the inputs that we used to have, and we've never watered our piles at Dignam Street before, but we will need to keep a better eye on our moisture. And so that's one thing that you look for. But as you saw, you know, like this, your eye's not always a great indicator. Like in the past, I would have gone, oh, there's going to be nothing in there. It's just wood and sticks. Doesn't look like it's broken down at all, but you just saw what we saw. and it's full of the biology. So, but you can do like a jar test of putting some in a jar with water, shake it up, let it settle, see what color that liquid is. And yeah, if you've got kind of honey brown, if you've got like beer color, you're in your fulvic acid phase. If you've got dark chocolate cacao color, you're in your humic acid phase. If there's humic acid, there is fungi in that, which so you can definitely do that as a visual assessment for your pile. So yeah, moisture content, do a jar shape for your color, and you can kind of start to see the biology. There's different indicators. You know, if you've got lots of slaters, it's a bit dry, but do dig in, because we've found we've got this sort of sacrificial layer on the top that looks quite dry, but dig in and you know, assess your moisture from further down than just your top and edges. Should you turn compost? I have both a static and tumbling compost. One does produce a healthier compost. Yeah, I mean, the less turning, the better. So we do 2 turns at the outset with this particular methodology, but if you're doing static piles, awesome. If you can keep your air in your static pile and it doesn't go anaerobic, that's going to be giving you really good fungi. So I mean, nature doesn't turn itself, well, it does with the microbes, but you know, you will get really good fungal dominance the less you turn it. We had a pile that I was checking through the microscope, we had really good fungal strands in it. We turned it because it did get a bit waterlogged, and so we wanted, and it had lots of indicators of that it was anaerobic through looking at it, and there were lots of ciliates, which is an indicator of anaerobic conditions, no moisture, no air. So, excuse me. We had good fungi before we turned it, and then the fungal strands went from being like big ones that we just saw now through to tiny little. It was definitely indicative that turning broke up those fungal strands. Now they will regrow, but the less you disrupt the system, the better it is for your fungal growth, definitely. So if you build a good static pile, then that's definitely a better option than a constant tumbling compost. You're going to be breaking those strands up every time you tumble it. Are worms a good indicator of soil health? Yes, definitely. So the worms we looked at here, the nematodes we looked at, you can't see with your eye, but yeah, worms are definitely a good indicator, and they are going to be they're part of the poop loop. They're going to be chomping up all the nutrients, pooping them out in a form that the plants can take up. Actinobacteria is an easy visible indicator of low oxygen. Sophie, yes, that's right. What does it look like? White, yeah. You guys are answering each other's questions. Awesome. Yeah, that's right. The actinobacteria often sits on, coats your woody material, and it is white. Sometimes people misdiagnose that as being mycelium, but I've checked it, and yeah, it's, when you look at it through the microscope, we didn't see any today, but it's very thin, it's colorless, and it's actually a long chain of bacteria rather than fungi. And it's an indicator that the oxygen level has dropped, is dropping below, I think it's 6 parts per million. And it's your pile's about to go anaerobic. So you can kind of see the actinobacteria on the top. And that's sort of an indicator that, yeah, in that situation, some actinobacteria is fine. It's actually great for brassicas. They grow really well with actinobacteria. But yeah, it's indicating that your oxygen's being used up. And so you might want to punch chimneys in your pile. So that's one way of not turning the whole pile, but it's getting some oxygen in there. So you can just, even with a broom handle, just shove a broom handle and give it a little thing, and that's going to be creating more oxygen to more surface area within your compost itself. What about ants? Ants, like, they're just consuming, really. I don't mind ants. They're just part of it, and often through a thermophilic step, you'll cook them off anyway. What's your thoughts on ants, Tim?
Yeah, the same. It's essentially, that means that there's a situation where it's dry enough and there's some food that's accessible and you haven't moved the pile in a while. Other than that, unless you really don't like ants, there's not really any problem per se.
Yeah. Well, that's awesome questions. Thanks, everyone. We're over, just over time after 11 o'clock. We would love to, if you want to join our email group, we'll be doing these as we're going through our testing. So if you'd like to get in touch and be on our email list, you can either go to Dignam Street Community Garden Facebook page, where we'll post some, you know, any of our events that are coming up, but I'll just post Ola's email address in the chat, and she's beautifully managing all our comms and our weekly email out. And so if you'd like to be on our weekly email list to find out when we're doing more microscopy or other cool stuff, thank you all for coming. Come visit us at Dignam Street Community Garden. We've got regular sessions on Tuesday mornings, 9:30 till 12:30, and we've just started a new Sunday session from 4 till 6, and come and have a look at our compost. Awesome. Thanks, everyone. Enjoy your day.
Got a bit. Thanks, everybody.
Thank you.
Awesome.
Have a good day, guys.
Thank you.
Thank you very much.